Here are some of the questions about microarrays, bioinformatics, gel electrophoresis, or DNA profiling that have came in wbi15 past papers. Obviously, this will not be enough. You need to study the syllabus content or the topic from the book. Then you would refer to these to know the keywords. Describe how microarrays in bioinformatics could be used to investigate the genetic basis of the Parkinson's disease. You'll have microarrays. You will define basically each microarrays, then bioinformatics, and mention the role of each one determining the genetic basis of the Parkinson's disease. Like here, microarrays will allow the identification of actively transcribed genes. We know this, right? And we know that bioinformatics, it's the computing tools or computers or softwares that will analyze, organize, and store biological data. Saying the definition of both will score you 2 out of 4. Then you will explain the role where microarrays will identify or analyze the activity of many genes in one single sample. Like in this single sample, you have several millions, hundreds, thousands of probes. This would identify or analyze the genetic activity of many genes. You collect information about genetic differences from many individuals, with or without Parkinson's. We know that in microarrays we compare the gene the genes activated for both the diseased with Parkinson and the healthy. Whether whatever the disease, it's always comparing the diseased genome to the healthy genome to know the genes activated here and there and how they're different. There are some genes that are activated in both, some are activated in each individual one, some are not activated in both, activated in neither, we can say. So microarrays will help collect information about the genetic differences between the large number of individuals with and without the Parkinson's disease. What about bioinformatics? Bioinformatics would help identify the differences in the base sequences of the healthy and the diseased with Parkinson. Explain how microarrays in bioinformatics can be used to identify the food toxicants that alter the gene expression. This was a question in article, so the questions in articles do not come that direct. So this was the actual question, and this is my prompt, or how will it come if it was an actual question from the first 7 questions. An important point we just need to discuss here that how will I know that the question in the article will be asking about microarrays or bioinformatics in general, or even DNA profiling? It's just from the context of the paragraph where you would see that they used microarrays, or that there are some genetic differences, or that there are some genes activated, or the use of bioinformatics, it's mentioned in the paragraph, and that's indicating, and that indicates the importance of preparing or reading the article previously before the exam. Anyways, let's start. How microarrays and bioinformatics can be used to identify food toxicants that alter gene expression. You use microarray, and details about this process or procedure. You would extract the mRNA, convert it to cDNA by reverse transcriptase. This is a qualified detail. And then you use bioinformatics, which is like the algorithm or computer or software that would analyze, do three things: Analyze, organize, and store. Obviously, the question won't come like this if the marking points are using, like here in this question, he gave a mark, 2 marks for only saying use microarray and use bioinformatics. So obviously the question won't come that direct. If it came like that, if it came not that straightforward, it came so vague, you must mention the straightforward points first, then you can elaborate according to your marks. Explain how the scientists could determine the high degree of homology. Homology, it's we know that homologous and or no, homozygous and heterozygous. Homozygous meaning that we have same alleles, both dominant, both recessive, AA or AA. This is a homologous. Homologous means same. Homo means same. So, degree of similarity of genes between humans and zebrafish. Obviously, you're going to carry out DNA profiling or DNA analysis using gel electrophoresis, where you can mention details about the PCR and amplification of the DNA. Then you would use the after the amplification, you'd use the gel electrophoresis. Always gel electrophoresis. It's following PCR. Obviously, you'd compare the base sequence, or the more the similarities between the base sequences, the more closely related the species, humans or zebrafish, are to each other. The more closely related, the more common the more recent the common ancestor is. And also, we must use bioinformatics to identify these genes. Plus, the last point would be that you must show that the products of genes have similar functions, meaning that the proteins coded by the genes are just having the same function, not having same or similar, not an entirely different function. They both contribute to the same function in the 2 different organisms. Explain how microarrays and bioinformatics software could be used to evaluate changes in expression in the here also this question was a was article. Here this too, like we have 3 article questions, so that indicates that articles target this topic. If it's not found in in the first 7 past paper questions, they'd be found in article for sure. Gut microorganisms produce LPS. White blood cells have receptors for LPS on their cell surface membrane. Describe techniques that could be used to identify the LPS receptor gene found in white blood cells. We must extract the mRNA from the white blood cell. Then we convert it to the cDNA using reverse transcriptase, and you must use microarrays. Where? We know that they sample or they analyze the activity of many genes in a single sample, and they would and we would leave the something we also forget always about the cDNA is that we must label the cDNA with fluorescent tags. This is an important point. You label with fluorescent tags, then the cDNA, then use microarray where the cDNA will hybridize with the probe, and then you would collect data from the microarray analysis or gel electrophoresis. Here, this is an alternative answer, okay? Go down both routes and take highest score. Like, there are somehow alternative answers, but I just disagree here because cDNA is the same as extract mRNA. Because, like, here, if we extracted mRNA, we would convert it to cDNA later on. So, these are both in the same route. However, the rest of the answer would be like diverting. It's like they're different. completely different. Refers to the use of bioinformatics to analyze the data. Obviously, you have come to know that just saying microarrays and defining them, or saying bioinformatics and defining them, would score you highest marks. Whenever you mention these two, you must define them, then mention their role. It's as simple as that. How microarrays and bioinformatics software can be used to identify genes that are actively transcribed in a specific tissue sample. This was so easy. Here you described, but here the point was that, yeah, this is the question itself. It just went down, but it's actually this one. Its answer is this one. This is the prompt of the article question. Here, let's come to the next question, which how you can identify the actively transcribed genes. In this question, it was 2 marks, and it didn't need the use of the bioinformatics, just was sufficient by the explanation of microarray procedure, and that was like the first time to do this. And I remember that this was a recent year, so many people lost the 2 marks, or lost actually the second mark. They had the mark for microarray, but they expected it to be like use microarray because it it samples many. It analyzes the activity of many genes in one sample. It allows it forms cDNA, and they just continued explanation by informatics. But he didn't need that. He needed the explanation of the procedure itself of microarray. So to be always on the safe side, to always explain the procedure, define the term, and link its role to the specific question. So be specific in its role, and fulfilling these 3 rules. I'll write them here. Define, then explain process, explain role. This is for both microarrays and bioinformatics in any question asking about DNA profiling, or like sequencing or stuff like this. Mutations in other genes can result in blindness. This question was talking about a gene, I don't remember, it was REP something 6 blah blah blah, and the question talked about that the mutation in this gene would result in blindness in a way or another. The question also discussed the possible treatments where we use gene therapy, stuff like this. But anyways, this was the last question in the last part in the question, and it was talking that mutations in other genes, like genes other than the gene I told you about, could result in blindness. How these genes can be identified? Obviously, we use microarrays to identify the actively transcribed genes where in the eye, because here we're talking about blindness. So you must apply to the questions topic or questions bulk, and always be specific. Then here, this question used like different keywords we weren't like used to, like sequencing. Sequencing was here the first time to be used. And actually the last time it wasn't, or like, to not lie, it's supposedly not the last time, because sequencing is in your syllabus, but it wasn't that frequent in questions. In that topic, in the topic of DNA, the more frequently asked topic was microarrays and bioinformatics. So here it was the first time to ask about the sequencing of genomes of individuals with inherited forms of blindness. Sequencing meaning that you identify the base sequence. But basically, it's better to write sequencing of genome. So sequencing of genome of the individuals with inherited forms of blindness, meaning that you just line up their order, or you identify the order of bases of in the people with inherited forms of blindness that inherited the blindness. Why we mentioned about inherited forms of blindness? Because the blindness, he said in the question, is due to mutation. And we know that mutation in the genes will result in a different base sequence, and this different base sequence can be inherited if it was present in the gametes. And also, this form of blindness was mentioned to be in RP blah blah blah. It was mentioned to be inherited, an inherited form. Then you would compare the sequences of active genes. Now you identify the sequence. Here in that step, you identified the sequence. Now it's time to compare sequence of active genes. Obviously, active genes, because that's what I previously said. I said in microarrays that they identify actively transcribed genes. These active genes are from a large number of both the blind persons or like individuals with blindness or without blindness to identify mutation. And we know, we have discussed this point before, that we need to compare the genome sequence with people, like between two different peoples, the two different groups of people, the diseased or the diseased and healthy, because we need to identify the genes that were transcribed in the diseased, so they may have played a role in the presence of the disease, and also the genes that are activated only in the healthy, so maybe they were silenced and then caused the disease in the diseased. Questions here about DNA profiling. These were direct questions. This was a question in an article in June 2024. And that's the prompt, the actual question. You can find it in that year. Explain how scientists proved a protective gene was transferred from a plant into an insect genome. You must determine the base sequence or the genome sequence for this insect, which was the whitefly, and you use microarrays. Then you use bioinformatics. You can define each. Bioinformatics was the software or computing tool or the algorithm to organize, analyze, and store biological data. Then you compare the DNA sequence of whitefly with the genomes of the plant to see if they were the same. This is to know whether they were transferred to the plant or no. Then this gene, if it's only found in the plants and not in any other species of whiteflies, so it had been transferred from the plant. So again, I have an insect from a population. It carried this gene. If it's the only one to carry this gene in its population and the rest of the plant population carried this gene, so that means it was only transferred to that species. Sorry, it was talking about not another species of whitefly. So it was this genome was only found in one species out of 10. So 9 species were without that genome, and one species had that genome that totally would support the hypothesis of the genome only present in plants, especially when it was only found in the whole plant species. Last thing is an advice. If a question asks about gene expression or the active genes or genes responsible for blah blah blah, genes responsible for Parkinson's disease, genes responsible for blindness, you would mention microarrays and bioinformatics. If the question asked about identifying mutations or comparing species codes, you compare species, you need to know the homology, the degree of homology between two species. You want to see if they're similar or not, they share a common ancestor, they are closely related. So that's all going to be answered by genome sequencing. The question is asking about finding a physical gene fragment or amplifying it. You felt that it needs amplification. You collected a small sample from the DNA, so you use PCR. Also, you can, these two questions are kind of overlapping, so you can use PCR and gel electrophoresis if he asked you about mutations or relationships between organisms or like evolutionary races. Evolutionary relationships. Evolutionary race was in the unit for